Welcome back to Cancer Sequencing 101. In our final episode, we're looking at the full structural architecture of cancer genomes, phasing, repetitive regions, and structural variants. Let's start with phasing. We all carry 2 copies of each chromosome, one from each parent. Short-read sequencing often collapses these into a single mixed sequence, losing the distinction between them. Phasing is the process of assigning variants to their haplotype of origin, figuring out which copy each one sits on. And to do that, you need reads that are long enough to span multiple mutation sites and then accurate enough to reliably call each one. HiFi reads check both boxes, typically 15 to 20 kilobases long and 99.9% accuracy. In cancer, phasing plays a critical role. Let's look at this PTEN example. In normal tissue, you can see both haplotypes clearly. But in the tumor, one haplotype is completely gone. That's loss of heterozygosity. Without phasing, this would just look like lower coverage. But with phasing, you can see that an entire copy of a tumor suppressor gene has been deleted. Phasing also determines whether 2 mutations on the same gene are on the same allele. That's in cis, or if they're on opposite alleles, in trans. In breast cancer, researchers studied patients with 2 mutations in the PIK3CA oncogene. When both mutations were in cis, patients showed a super responder phenotype to the targeted therapy alpelisib. But when the same 2 mutations were in trans, that enhanced response disappeared. Short reads can entirely miss this distinction. They can tell you the mutations exist, but not which chromosome they're on. Next, let's talk about repetitive regions. These are sometimes called the genome's dark matter. Tandem repeats make up a significant portion of the human genome, and these regions are linked to dozens of diseases, including cancer. The problem is that many pathogenic repeat expansions are hundreds or even thousands of base pairs long, far beyond what short reads can span. They pile up and they collapse during alignment, making accurate genotyping nearly impossible. But HiFi reads solve this by spanning an entire repeat expansion in one single read. And PacBio's Target, the tandem repeat genotyping tool, then genotypes each expansion with single base resolution, separating the maternal and paternal copies so that you can see the length, sequence composition, and even the methylation status of each allele. Just look at the difference. In normal tissue, both copies are short and stable. But in the tumor, one allele has undergone a massive expansion. There are hundreds of extra copies that could otherwise be invisible to short reads. And finally, let's talk about structural variants. Large-scale changes to the genome, like insertions, deletions, inversions, and translocations. Structural variants are one of the main drivers of cancer, but here's the problem. The vast majority can be invisible to short reads. They tend to lie in repetitive regions, and they're often larger than the short reads themselves, so short read sequencers really easily miss them. But HiFi reads change that. Because they're long enough to span the entire variant and read into the unique flanking regions on either side, you get clean, accurate detection of the breakpoints, exactly where the rearrangement happened. The difference is dramatic. In the SKBR3 breast cancer cell line, a model for HER2-positive breast cancers, researchers found that the vast majority of structural variants were undetectable with short reads. And in a study of pediatric B-cell leukemia, many patients had no identifiable driver mutation using standard tests. They were classified as not otherwise specified. But when those same researchers applied HiFi whole genome sequencing, that unknown category shrank dramatically. HiFi detected translocations that the standard tests had completely missed. In one case, it revealed a fusion between TCF3 on chromosome 19 and ZNF384 on chromosome 12, a specific subtype that can potentially impact a treatment approach. HiFi doesn't just read the genome. It reveals its full architecture. To explore all the cancer biomarkers we've covered in this series, visit our interactive cancer body map. The link is in the description.